All right, I think we can get started. Welcome, everyone. This is our public webinar series about the Common Fund data ecosystem research that we at the Knowledge Center are hosting. So I'm Maria Costanzo. I'm on the Knowledge Center team at the Broad Institute, and I'm filling in for my colleague Mackenzie Brandis, who's out today, who normally does this. So before today's presentation, I'll just start out with a few little updates, scientific updates, and talk about some upcoming events, and then we'll go on to our featured speaker. So I just want to remind everyone that these webinars are all recorded and they're available on our YouTube channel, which is CFDE Knowledge Center. We have 6 of them so far, and so you can go and catch up on any that you missed there. Also wanted to remind you that our team is working on the Knowledge Center portal. And what we want to do is represent results from all of the Common Fund DCCs, data coordinating centers, and make it useful to researchers everywhere, not only within the CFDE, but anywhere in the world. So if you are a researcher who is using data from one of the CFDE resources, we'd love it if you would contact us to talk about how you use it, because that would be really helpful in us in thinking about how to represent these results. And yes, please stay in touch. This is our help email address, so please email us with any suggestions or questions, or also if you would like to be added to our email list. So some upcoming events. We are going to be attending the American Society of Human Genetics Conference in early November in Denver. And we'll have an exhibition booth that's all about the CFDE. We're also hosting an ancillary session that will have presentations about CFDE research. This is still a bit in the planning stages, but if you are going to be attending ASHG, please do let us know, because we'd love to connect with you there. And we'll keep you updated about the details of what's happening there as that develops. Another upcoming event is that our next webinar, a month from now, is going to be a presentation from the MotorPack consortium. And so today, we are having a presentation from the Extracellular RNA Consortium, which is one of the programs in the Common Fund data ecosystem. And our presenter will be Keyang Yu, who will talk about tools and methods for discovering disease-causing gene regulatory variants from whole genome sequence data with application to congenital heart disease. So thank you for being with us, Keyang. I'll stop sharing and you can share your screen.
Sure. Thank you for the kind introduction. Let me share screen. Okay. Can everyone see the slides?
Yes. Yes.
Okay. I see a hand raised in the chat. Maybe we should.
Oh no, I just hit it by accident to let you know. We're good.
Oh, okay, I see.
Sorry, we're
good.
All
right, nice. Okay. Oops, sorry, that's not the slide I want to start with. Okay, I'll start my presentation then. So as the introduction says, today I'll be presenting on the work of xRNA DCC, in particularly in collaboration with with Kids First and the GTEx DCC on the GeneRec partnership. My talk is going to be on tools and methods for discovering disease-causing gene regulatory variants from whole genome sequencing data with application to congenital heart disease. So our project really builds on the basis that the increased accessibility of whole genome sequencing allows us to perform more large-scale studies of non-coding genome. And one of the most common ways to study the non-coding genome is through GWAS studies, or genome-wide association studies. Based on a review article from 2021, we currently have more than 5,700 GWAS studies for more than 3,000 traits, and the push for more statistical power has thrust the sample size to well beyond 1 million participants. The mass majorities of these GWAS results, which hits the single nucleotide polymorphisms, identified are localized in the non-coding region. And a significant fraction of those variants impact the transcription factor bindings to enhancers and promoters and such. That being said, GWAS studies have its limitations. If I may direct your attention to the figure on the right, most of the variants hit by the GWAS studies land somewhere in the middle section of this figure. So they're either high penetrant mutations, they're intermediate frequency variants with moderate effects, or common variants with small effects. And they usually falls on the more common allele frequency range. So on the right-hand side of the line in the middle. So most of those variants has relatively high frequency, and it's hard for GWAS to pinpoint rare causal variants that are in a patient. In order to help with the study of rare variants, such as de novo variants and ultra-rare variants, we created this framework for discovery of pathogenic rare non-coding variants. And I know the figure on the right is kind of a summary of all the components of our framework, and I'm going to go through each one of them. So the most important part of this framework, and which is the heart and core of this framework, is the point-wise mutual information models. And what is point-wise mutual information? This is a measure of how two things happen together versus how often we expect them to happen by chance. This is most commonly used previously in natural language processing. We're looking at when two words or two phrases are going to happen together. In this case, we're applying the point-wise mutual information system in the study of rare variants. How do we do that? Now, given a condition C and genomic position X, the information theory provides us with a p-value for de novo variants at X that we denoted as p-PMI. Based on the mathematical nature and symmetry of PMI, as well as Markov inequality, we then reach the equation that this informatic theory p-value, p-PMI, is smaller or equal to the possibility of X, so px, over the possibility of X given C. So the possibility of a variant happening in a genomic position C given the disease condition C. This framework then provides an equation, provides us with an ability to make use of phenotypic data and epigenomic data from different databases to refine this px given C and improve and optimize the p-PMI. And allow us to create different conditional probability distributions for different disease subtypes to address heterogeneity of diseases, which I'll address on a later slide. The first key question, however, is how do we aggregate regulatory information to aid this building of PMI models? Most of the regulatory information epigenomic regulation out there right now are separated in different data silos and cannot be easily accessed together. To do so, we've built a fair infrastructure that links gene regulatory data, information, and knowledge together. I'm going to mainly talk about 3 databases that we have built to aid this effort. The first two, very similar to each other, are the Congenital Allele Registry and Genomic Location Registry. These are 2 services that provides stable and canonical identifiers for variants, genomic positions, genomic locations, anything that is in the genome. This particular entity is mapped across the genome references and transcripts, and new identifiers are always available on demand via either a web UI or an API endpoints. So this allows people from different expertise to talk about the same entity across different fields of studies within a study of genetics and genomics. Now we have this identifier that allows us to talk about something. It gives us the ability to aggregate evidence for regulatory relationships about these variants. And we collate these regulatory information using the CFDE Link Data Hub. The CFDE Link Data Hub is a graph-based database that links excerpts of information from different databases. The purpose of Link Data Hub is not really just another place to hoard information, but we want to connect the different databases. So we also provide links to the original source of information about those variants. All the documents within this database, this Link Data Hub, is structured such that it is standardized using ontology terms. They're permanent, they're versioned, they're also API accessible in machine-readable or human-readable form. Essentially, we want to use the CFDE Link Data Hub to create a PubMed, so to speak, for regulatory elements and variants so that information can be used as supporting evidence in studies. This is a simplified structure of the CFDE Link Data Hub. I'm not going to go through each part of them, but just want to highlight 2 things. One is if you look at the color-coded parts of this graph, we have a red section, which is the information section, and the blue section, which is the knowledge section. The information section are examples of it would be EQTLs or SQTL. So these are results of data processing based on specific methodologies. Knowledge, on the other hand, are assertions supported by different evidence that are not tied to any specific methodologies. So you could think of it as a summary of different sources. The second notable things I want to point out in this particular structure is everything within this linked data hub graph is linked to a context collection. This can be a tissue or cell type, but essentially the context of specific studies or specific statement being made. CFDE linked data hub is currently linked to regulatory information from more than 10 different sources, covering most human coding genes. You can take a look at its statistics, but I only want to point out two things. One is we have regulatory information from screen database. So this is a cis regulatory element database based on ENCODE data. We also from allele-specific epigenomic signatures from the NTEX and ROMAP studies, as well as EQTL and SQTL from the GTEX studies. Other information such as NOMAD, allele frequencies can also be found in the linked data hub. Now that we have these regulatory information excerpts in one place, it helps us to build different PMI-based probability model based on this tissue and cell type specific structure. How this is done. So I just want to show one example where we're building a cardiomyocyte-based PMI model. In this case, we take all the cardiomyocyte-associated tissue and cell types from CFDE linked data hub, and we queried for evidence and knowledge associated with that particular context. We then would be able to get regions, for instance, that are active in cardiomyocyte-associated tissues or cell types. We then, based on the PMI, upweighted the probability of a rare variance or de novo variance at a particular genome position being upweighted in this condition. We then compare this PX given C with PX to get the PPMI for each patient based on this PMI model. This then helps us to identify a rare index variant for each patient or rare ring var. Now, we first apply our PMI models in a sample or a primary use case of congenital heart disease. It is one of the most common congenital disease in the U.S. with a high occurrence every year. Previous studies of de novo variants of the variants that appears in congenital heart disease patients show that only a small portion of these have known causes, and only 8% of this are contributed by coding de novo variants. This therefore tells us that there's a high probability that non-coding variants contributed to congenital heart disease as well. In this case, we're using data from the Case First database, where we'll have around 1,000 congenital heart disease cases from this publication that makes use of Case First data. We also use as a non-congenital heart disease control from the same publication. The de novo variants from these individuals can be obtained through first the Case First Data Resource Center, which enables data file browsing and selection in a particular cohort. These files can then be automatically transferred from this DRC UI to Cavatica for further processing and filtering. We therefore were able to gain de novo variants through this variant workbench that Case First developed. Using this de novo variant, the relative information we obtained, as well as a curated list of CHG-associated genes, we're able to perform a probability study that finds, when comparing the p-value PPMI of the DMVs from the CHG cohort and control cohort, first, there's a significant shift towards smaller PPMI for the CHG cohort variants. Second, there's an enrichment based on rank sum tests. There's an enrichment of CHD cohort high p-value variants within small p-value range. This allows us to identify a rich set of ring var in CHD cohort based on this cardiomyocyte model. When comparing the cardiomyocyte model with the general model, which makes use of not only cardiomyocytes, which does not focus on cardiomyocyte-specific relative information, we see a significant increase in not only the enrichment ratio, but also the rank sum test results. This shows that enrichment or identify of rare pathogenic variants is very dependent on the right information as provided by linked data. Down the line, we also want to explore the tissue and cell type specificity matching with the different subtypes of congenital heart disease. In collaboration with Dr. Lili San, Dr. Andrea Wilderman, and Dr. Shane Morris from Baylor College of Medicine and Texas Children's, we identify 5 major subtypes of congenital heart disease found in the clinical settings. them being left ventricle outflow tract defects or obstruction, canonical defects, laterality, atrial septal defects, and right heart defects. And I've listed a number of these cases within the kids' first cohort. We want to use our models that are being built in different tissues and different cell types and match them to the different congenital heart disease subtypes to identify rare variants in a more accurate way. All these regulatory information analysis tools are currently publicly available by either UI API or Cavatica app. First, we have the CFDE link data hub, which is accessible through this link on the slides. This provides you a chance to use the UI to identify human-readable information, but the same information is also available through API endpoints, and JSON documents can be captured for machine-based analysis. We also, just a sneak peek into the future, we're also developing a possibility for you to own your own instance of Link Data Hub so you can explore it or develop it as you want. The PMI models, the cardiomyocyte one and the general one, are also currently available on, should be available on Cavatica soon. We're working with the Cavatica team to make this model be publicly available through public projects and app. The regulatory information from CFD Link Data Hub is also accessible through Workflow Playbook. This is through a collaborative effort with the Playbook Workflow team. Is through especially with Avi's team. And this allows you to create workflows that discover the regulatory information about a particular gene, regulatory elements, or variants. In this case, I'm just showing a workflow that I've built that helps you to identify associated allele-specific epigenomic signatures to a variant. So all these resources are available and freely accessible to external users, and I'll take any questions.
People can put questions in the chat or in the Q&A, or you can raise your hand and we can unmute you, whatever you like.
I guess I'll ask a question about the playbook, Keyang. Can you do that? Can you run the playbook for other phenotypes within Kids First, or just for the congenital heart disease? Or is this just about the variants? What can you determine with the playbook? Because I can imagine it's very useful by other people.
Yeah, so for the playbook content, really we focus on the central entities, which are the genes, regulatory elements, and variants. So if the variants or if any of these entities are in Link Data Hub and query about them and build a workflow about them in playbook, you'll be able to get any regulatory information associated with these entities.
I also would like to add that the app will be available in private workspaces on Cavatica for use on protected whole genome sequencing data.
So that would be where, Alex, you could query different types of phenotypes with the exact same framework that Keyang has built, correct?
Exactly, but
that's excellent.
At this time, we're starting with congenital heart disease and one subtype of it, which is LVOT, which can be explained by variant effects that are cell type autonomous, cardiomyocyte specific. And so the model that we developed is really optimized for that subtype. But we are developing other models for other subtypes of congenital heart disease, and we're going to use this framework, hopefully in the future, to expand, to build models for other congenital diseases. But the key to that is really aggregating information about gene regulation, and then building those models based on all current aggregated knowledge so that we get that specificity that we got for LVOT.
Got it. Very good. That's excellent.
So we envision a library of these models that will be versioned and evolving. And the app can work in a private workspace on Cavatica or other cloud platforms such as Anvil, where the app will be run locally and it will fetch, the model from a model repository that's API accessible. So there won't be any leakage of data, from the private workspace. We envision that this setup will allow, aggregation of knowledge that's public and then deployment in a private setting, as well as flow of information in the other direction. Because we only identify one rare index variant per individual, that one variant and its p-value can actually be shared publicly, similar to ClinVar serving as a repository for the sharing of pathogenic variants. In contrast to pathogenic variants, these variants are not established to be pathogenic necessarily, but they may point to regulatory elements, genes that may be relevant for a particular disease. So in other words, this is more of research type information as opposed to clinically valid information, but still it can be shared. So we envision this kind of bidirectional sharing of knowledge information to allow us to shed lights on the role of regulatory elements in various congenital diseases.
Excellent.
Any other questions? Feel free to put it in the chat and I'll read it. Oh, I think I see attendee.
Have a raised hand.
Yeah.
Let's see if I can unmute her. Gosh. Sorry, I'm not a Zoom expert. Oh yeah, okay, I should be able to do it. Sorry. Let me find Julie in the, okay, allowed to talk. Okay, Julie. You should be able to speak.
Oh, can you hear me?
Yes.
Yeah.
Hi, sorry about that. This was really great. I think this is an exciting model. How much applicability does this model have outside of the scope of rare de novo variants? So for instance, if an individual is suspected to have an autosomal recessive disorder, would you be able to integrate this model or a version of it for identifying those types of variants as well?
I believe so. It really depends on kind of what information we are feeding the PMI model, kind of what type of model we build. But I don't think it's the PMI model is limited to just rare de novo variants.
Just to add to Keyang's response, this model in principle is applicable to ultra rare variants where extremely low population allele frequency is applied to filter variants. The advantage of de novos is that they're small in number, so on the order of 100. So the p-value that we derive gives us an idea about how many variants we can deal with, right? Currently, the p-value is on the order of 1 in 100. So we can handle about 100 variants with reasonable expectation that the most high scoring variant is not high scoring by chance. So if we go into cutoffs that are, for ultra rare variants, allele frequency based, then we may get maybe 1,000 variants. So to reach that informativeness, we may need a p-value that's on the order of 1 in 1,000, right? So we actually don't have 1 in 1,000 p-value based on a few hundred samples that we have. But as we accumulate more information, we can actually move away from the de novo requirement, and we can start working also with ultra rare, de novo variants. The key to this, of course, is to have very good, very low allele frequency cutoffs, so we don't have too many variants to consider. Allele frequency is not used, right, for our probability
Not
model.
currently. Mm-hmm.
So it is independently used parameter, and this model kind of sheds light on interplay between functional information and allele frequency information in allowing us, to discover variants. So in other words, as we accumulate more regulatory information, we'll be able to tolerate more variants per individual, and we can go away from the de novo requirement and consider the ultra rare variants as well.
Great, thank you.
Any other questions? All right, if not, I guess we can conclude. Thank you all for coming. We'll make this recording available on our YouTube channel so you can catch up, or anyone who missed it can catch up. If you have any other questions or comments, please do email us at our help email address. Okay, and thank you all for attending. We'll see you next month. Bye-bye.
Thank you.
Thank you.
Bye.